Good afternoon, ladies and gentlemen, and welcome to the fourth seminar in the series, Challenges in Forest Ecology. This comes to you from the Faculty of Forest and Wood Technology at Mendel University. Today's speaker is Professor Andrea Polle from the University of Göttingen. Andrea Polle was until recently Professor of Forest Botany and Tree Physiology. Her major interests are molecular ecophysiology of stress and adaptation in trees, as well as biotic interactions under globally changing environmental conditions. Much of her focus has been on tree mycorrhizal interactions, which is what we're going to hear about today. Andrea Polle is a leading scientist in the field of plant sciences. She's published more than 300 scientific articles and has been cited more than 34,000 times. She's an editor of the journals such as New Phytologist and Tree Physiology. She's affiliated as a visiting scientist to Beijing Forestry University, and she's been elected Fellow of the Academy of Sciences of Lower Saxony, where she's currently serving as Vice President. Well, today she's going to talk about mycorrhiza and the Wood Wide Web, facts, fiction, and uncertainties.
Thank you, Douglas, for your kind invitation. I'm very honored to be here today and hope that I can tell you something new and stimulate a discussion and perhaps also some thoughts. The topic today is mycorrhiza and the Wood Wide Web, facts, fiction, and uncertainties. And I've chosen the topic because it's a highly discussed topic at the moment. The term Wood Wide Web was initially coined by Nature journalists. The cover page publication in 1997, so it's more than 25 years ago. And this was based on a seminal paper published by Susan Simmert as the lead author, showing that there is a translocation of carbon below ground involving most likely mycorrhizal hyphae connecting trees. This publication stimulated a lot of research because the flow of carbon into the soil is really one of the fundamental processes in soil ecology. The amounts of carbon which may be sequestered play an important role also in the current discussion, for example, with regard to climate change. This initial paper stimulated a lot of research, and this is, of course, something very important in science, and especially in the recent years with the progress in molecular methods, it has become even a new, I would say this is going at a new pace. However, in addition to papers supporting these initial observations, and I will go into details a little bit later, there are also a number of publications which show that this topic is really controversial. And it led not only to scientific publications, but also to a lot of popular publications, especially best-selling books, for example, published by Peter Wohleben. Eventually, the whole discussion culminated in this publication published by Justin Cast, Melanie Jones, and Jason Zimmer, who scrutinized and investigated the number of publications, the citations, and they found that not only the number of publications increased, but that there was also a trend towards an increasing percentage of unsupported citations. And I think that this is really a critical issue in science, and it was good that this was brought to the general attention. However, as you can see here, and this is, in my opinion, really very, very unusual, this was taken up even by public media. For example, the Bild Zeitung, which is one of the major newspapers in Germany. They interviewed Peter Wohlleben, and he disqualified this publication as unscientific and a personal opinion of the authors. I would say that scientific publication makes it into a discussion of the Bild Zeitung really shows that the basis of this whole area of carbon sequestration, carbon sharing between trees has reached a popular status and is really a big topic. The best selling books which have been published and which are partly at least based on this topic are, for example, The Hidden Life of Trees. And these books are based, for example, on publications of Susan Simmert, who claims that trees care for their offspring by providing resources via mycorrhizal networks in soil. And Peter Wohlleben then widened the concept and said, trees and forest stands care for each other by below ground resource sharing. And this means not only resource sharing between, let's say, adult trees and their offspring, but also old trees, but also healthy trees care for sick trees or care for old trees. And at this point, I think it is very important to take a step back to consider a little bit more in depth what is going on here, and to scrutinize the evidence, what is in favor of these suggestions, and what do we have to reject. I was happy to be able to join a group of scientists, and we investigated the literature about this. We published our opinion about this. I think that this is a very, in my opinion, an important step to go, because we as scientists should provide solid information, and we should scrutinize the information. It is not our task to make decisions, but we have to give a sound advice to decision makers. And if certain claims are wrong, or at least have to be discussed, then we should make this very clear to the wider audience. And this is now the topic of my talk today. And I would like to take all of you on this journey. And since I do not know if all people are very familiar with mycorrhizal fungi, I will just start with 2 slides showing a little bit of an introduction to mycorrhizal fungi. So first of all, you can see here a cross-section through a mycorrhizal root tip. The green shows the mycorrhizal hyphae, which grow between the root cells and which form the interface for the exchange of nutrients. If you look at this from the outside, you can see here that a tight network-like structure is formed around a root tip. And for comparison, I've also shown you here a non-mycorrhizal root tip, where you can see the root hairs growing out of the root tip. In this slide here, you can show that starting from the mycorrhizal root tips, hyphae emanate into the surroundings, and they are important for the uptake and translocation of nutrients to the tree. Only a few words about the carbon physiology. All of you know that the CO2 is taken up by the tree, carbohydrates are being formed, and they are then translocated in the phloem to different zinc tissues. This can be storage in the stem, short-term and long-term. And this is an important point because we know that in the stem, even very old carbon, more than 10 years, for example, can be stored and later used under certain circumstances. Of course, the carbon is also utilized for growth. It can be retranslocated in xylem sap, and it is also used, for example, for the supply and flow to mycorrhizal fungi and for root exudates. Furthermore, we have to consider we also have stem respiration and that we also have stem photosynthesis. And these processes are important to consider when it comes to stable isotope techniques to label, for example, the pathway of carbon in the soil, because you can see that respiration, refixation will lead to mixed signals. Of course, it is important then also to consider the timescales under which these experiments are being conducted. How important is carbon sequestration in fungal mycelium in soil? I have just picked here a few, let's say, fairly arbitrary examples showing the wide range of numbers which have been published from 160 to almost 6,000 kilograms of carbon per hectare. An important point here is also to remember that the carbon stored in fungal biomass is not only due to mycorrhizal fungi in the soil, but part of this is also due to saprotrophic fungi. And the relative fractions of saprotrophic fungi and ectomycorrhizal fungal hyphae in soil may vary depending on, for example, whether we have conifer forests or deciduous forests. In my opinion, a very nice and very comprehensive overview on this topic has been published by Finlay and Clemenson, and they report that approximately 100 to 900 kilograms are fixed or are present as ectomycorrhizal hyphae in soil. And of course, you can also see here that not only the standing biomass is important or the pool of carbon is important, but also the turnover of this pool. However, these numbers also show that there is a fairly high degree of uncertainty and that we have little information on the contribution of different fungal species to these pools. So do all fungi do the same thing, or are there many differences among different fungal species? Because that could perhaps also explain some of these differences. In order to address this topic, we have recently published a study in which we exposed young beech trees, which were grown in their natural soil in mesocosms. They were colonized by their natural fungal communities. And we collected the mycorrhizal root tips, distinguished different fungal morphotypes, we sequenced them, and then we used these mycorrhizal root tips also to determine the isotopic signature in the mycorrhizal root tips. As a result, you see here that there is a lot of variation in the extent or the concentration of new carbon accumulated in these mycorrhizal root tips. For example, here we have Tomentella, here we have Xenococcum. This is also a Tomentella species. This is Pachyploides, and we have several not well characterized fungi. We know that they belong to the order of the Helothiales. We know that many fungi in this order are saprotrophs. However, here in this case, we also checked by cross-section that these fungi were really mycorrhizal fungi. You can see that the variation here is approximately a factor of 5 to 6 or even higher, suggesting that if we give a pulse of labeled carbon and investigate this after a chase period, in this case after 5 days, we see quite a huge variation of carbon which is accumulated by different fungal species. Interesting question is now, of course, I mean, what is the reason for these differences? We checked, for example, if the biomass of the fungi played an important role. This was, according to our data, not the case. But what we saw was that the ratio of CN of the mycorrhiza, of these ectomycorrhizas, was important. We found a very strong correlation between 13C, between the accumulation of 13C and the CN ratio. I think that this is an interesting and important result. It is not a mechanism, to make this clear, because it is a correlation, so we do not know the molecular underpinnings of this. But if we assume that the CN ratio is the driver of carbon into soil, the CN ratio of the ectomycorrhiza, then it also becomes clear that changes in the CN ratio will change the flux of carbon into the soil. And at a certain point, the flux of carbon can even stop. And this would be an important regulatory mechanism. Now, once the carbon enters the soil, it is found in mycorrhizal, or it's found in fungal hyphae. I'm sorry that this slide is a little bit, maybe very busy. It was an investigation conducted by colleagues in Göttingen from the soil science group, and they found using lipid biomarkers for certain microbial groups. So the G pluses here refer to bacteria, gram-positive bacteria, the G minus are gram-negative bacteria, SF are soil fungi, and you can see that the majority of carbon, of new carbon, which replaces 12 carbon in this marker lipid, is present in the soil fungi, in the group of the soil fungi. Please note that here the scales are, that the scales of these two are different. Once we have the carbon now in the soil and in the soil fungi, we come to the main topic, and that is the common mycorrhizal network, how this World Wide Web is nowadays usually called. We have to make a few definitions. So the first definition is, I mean, what do we really consider as a common mycorrhizal network? And most researchers, I think, would define this as a network which is formed of the same fungal species. It is not necessarily the same individuum, but the same fungal species between two different tree species. What we do not know is if, for example, this common mycorrhizal network can also integrate other fungal species, so that they would connect also with, so that hyphae of different fungi connect. And of course, we also have to consider that there are saprotrophic fungi in the soil. And now we come to the question, how important are these connections, and how much carbon can be transported from one species, from one tree species to another? And to make it, or to highlight this once again, we do not only talk about mycorrhizal or the translocation here from one fungus here to the next colonized root, but in this concept of resource sharing among trees, the carbon would have to enter the neighboring tree. I think it is worthwhile to go back to the roots of the whole story, and therefore I would like to show you the early experiments which have been conducted by Susan Simmert. So they had a microcosm experiment in which they grew 2 different tree species. The tree species, one was the birch, the other was the Douglas fir. They were encased here in, or the aerial space was either supplied with 14C or with 13C. And this enables them now to look at the exchange of carbon from one compartment here to the other compartment, and to quantify how much carbon from the Douglas fir would be present in the birch and vice versa. When they did this, they found that they found a net translocation from birch to Douglas fir. And now to control how much contribution is due to the hyphae, they cut the connections and checked then the amounts of carbon which were transferred. And you can see here, when they cut the connections, the transfer decreased approximately by a factor of 2. Black one, or the gray one here, is the total bidirectional translocation, and the white one is a net translocation. None of these differences were significant if we apply the usual 0.05 threshold for the probability. For the probabilities. I mean, if we take this now, then I would say the take-home message here of this experiment is that there is a translocation between the trees, and that approximately 50% of this translocation in this experimental setting can be assigned to hyphal connections. In their Nature paper, they used a similar strategy. They planted birch and Douglas fir, and also Taxus baccata in this case as a control. And then they checked for the exchange of carbon between the trees. After approximately, when they did this experiment, after 1 season or so, after planting, they did not find any effects. But if they looked in the 2nd year, and in particular, if they exposed the Douglas fir to shade, then they found a significant enrichment in the plants. And I think it's worthwhile really to look how they interpreted their results. They said, if our results reflect the magnitude of carbon transfer in natural system, then the net competitive effect of one species to another cannot be predicted without a better understanding of interplant carbon transfer through shared mycorrhizal fungi and soil pathways. And I think that this is a very important sentence, because it shows that the soil pathways can really not be neglected, and that is something which happens nowadays in, let's say, at least in some citations. Similar magnitude of carbon sharing was also claimed by a publication of Klein et al., which appeared approximately 10 years ago in Science. Fact as such that carbon is present in what we would call a receiver plant does not mean that the carbon is really present in the plant. It could also be present in the mycorrhizal fungi. And therefore, it is important to check how much of this carbon is translocated above ground. In this particular case, if I remember right, it was approximately 15%. So the overall amounts of carbon which were shared between trees was not very high. One possibility is, of course, that the carbon translocation works better between related tree species. than between tree species which are unrelated, phylogenetically less related. And that is symbolized here by this slide, suggesting that, for example, a conifer species would provide more carbon to a young conifer seedling and less to a deciduous tree species. So how much evidence is there for this idea and concept? Here in this study, Teste et al. Investigated the translocation of carbon between Douglas fir donor trees and seedlings using mesh bags. So either no mesh bag or mesh bags with decreasing mesh pore sizes to allow roots or hyphae or no hyphae to connect with the seedlings. And in this study, no significant enrichment was found, neither in the roots nor in the shoots. In another study, which appeared recently, a spruce tree was labeled, and then the translocation of the carbon was investigated between pine, larch, and beech, which surrounded this spruce seedlings. The authors then checked the similarities of the ectomycorrhizal community, and they quantified the carbon transfer. I will walk you here through this figure. So this orange shows the similarity of the fungal communities, and it means that the similarity between spruce and larch or spruce and pine is very high, and the similarity is less between spruce and beech. So the fungal communities differ more. This, what is it, violet figure shows now the carbon transfer, and in this case, they found also if the phylogenetic relationship was closer, between spruce and larch or spruce and pine, the amount of carbon was higher than between spruce and beech. And this would also support that that overlapping mycorrhizal communities is sharing a lot of different or sharing a common species pool can translocate more carbon among related tree species. And I think that would be an ecologically very important observation. However, whether it is important in terms of the mother tree concept that species which are more closely related supply their offspring with carbon to promote, for example, their growth is a huge, there we have a huge question mark, and I will show you why. So the first thing is that we have to quantify the amounts of carbon, and this was done in by Pickles et al. They exposed Douglas fir donor plants to 13C and then traced the signature in kins or non-kins of Douglas fir. So the black here is kin and red is non-kin, so less closely related Douglas fir seedlings. You can see that in this mesocosm experiment, part of the labeled carbon appeared in the soil. The soil of the receiver plant was separated by a mesh, and then the carbon was transferred to the soil of the receiver, to the root of the receiver, and to the shoot of the receiver. And here you can see that you cannot see anything. Yeah, and that is because the signal is really very, very low. If we now show this in a more, in a rescaled manner, you can see indeed there is a signal in the root of the, in the receiver soil, in the receiver root, and also in the receiver shoe. However, the amounts which have been translocated are really extremely small. Furthermore, they also conducted the experiment when hyphae were allowed or when hyphae were restricted. And you can see here the relative degrees of carbon which were exchanged between kin and in a kin and in a non-kin situation of the receiver. And you can see that in the permissive situation, the receiver, the kin receiver, received more carbon. However, when hyphal growth was restricted, they also received more carbon. And this speaks against hyphae being the main pathway of translocation, at least in this experiment settings, we have to say. Pickles and al. Interpreted her results in that way that there was root exudation, and that then the root exudates would be taken up by the mycorrhizal fungi of the receiver and then translocated. So the evidence for or against significant amounts of carbon which are being transferred is relatively, let's say, is relatively weak. The question now is what is now left? If we look at this concept, we have the donor trees, we have translocation and significant contributions of mycorrhizal mycelium to soil carbon. However, the amounts of carbon received by seedlings are to all what I would say very, very low and cannot contribute and cannot or do not have an ecological significance for these seedlings. We also have to consider that in the soil in the situation in the forest, we have all these other transfer pathways like respiration, exudation, turnover, re-assimilation of the carbon, and the redistribution by soil biota. And I think that this is often overlooked, that these networks are not, or that these networks are very dynamic, because Polyombola feed on them, earthworms feed on them, and so on. Yeah, wrapping up at this point, I have shown you that the evidence of the significant carbon transfer via common mycorrhizal networks that benefits the recipient plants is lacking. Generation patterns do also not support that mycorrhizal hyphae play an important role in this regard, because trenching experiments have shown that trenched regeneration, so which is disconnected from the mother trees, grow better than seedlings which grow in a non-trenched situation. The fungal pipeline connecting trees for net carbon to the host as a receiver is difficult to reconcile with an adaptive advantage for the fungus. So this means if the carbon is translocated from one ectomycorrhiza to the ectomycorrhiza attached with another tree species, this may be an advantage for the fungus, because if the fungus is connected with various tree species, it can always feed on carbon if it still can feed on carbon if one of the trees fails. However, what would be the advantage of the fungus to provide the carbon now to a receiver plant? This, of course, it is possible that carbon is transferred to a receiver plant. For example, we know that this situation exists for microheterotrophic organisms. However, in this mycorrhizal situation, it is difficult to see. Because if, for example, we assume that the carbon would be translocated together with nitrogen, for example, in the form of amino acids, then we would expect that the tree would have to provide carbon skeletons to the mycorrhiza to produce the amino acids. And this would mean we would have an exchange of carbon and probably no net gain of the receiver. Therefore, the question of an ecological significance is open at the moment, but we have only very few papers which support such a mechanism. With this, I'm at the end. I would like to thank you for your attention. I would like to thank Douglas, not only for the kind invitation, but also for bringing me in touch with mycorrhizal fungi that started when I initially came to Göttingen, and Douglas was still working in the Forest Botany Institute and doing mycorrhizal experiments. I would also like to thank Rodica Pena, who is now at Reading University. With her, I collaborated for many years. And special thanks also go to Christian Ammer, Nielsen, Tony Nesholm, and David Robinson for pushing me also to contribute to these publications, where we try to sort a little bit where we try to sort the importance of mycorrhizal networks compared, let's say, with the mother tree concept. And of course, apologies for many studies which I could not mention in the frame of this presentation.
Excellent. Thank you very much indeed. So we can take questions now. I'm sure there are scientific questions and there are science philosophy questions attached to this. General life questions, I would say. The first question is from Dave Johnson, which is your final point. Yeah. A benefit to the fungus in supplying carbon would be securing a potential long-term supply of carbon, even if the initial phases of host plant receiver growth are a net sink. There is evidence in green orchids of this give now, pay later scenario.
I think it's perhaps more a comment. I agree that this may be a mechanism. However, I would say in temperate or boreal forests, we do not have evidence for this. Because, I mean, if the young trees would really benefit from this, or if young trees would really benefit from this, then we would expect that if we disconnect the young trees from the mother trees or from these hyphal connections, they would not grow. They would not grow very well. Whether the fungus can, let's say, or whether organisms have a strategy for, let's say, very long time periods until they would perhaps benefit from, let's say, donating carbon, like maybe insurance or something, for a very far future. I have some doubts on this because we really talk about very long time scales in this regard. And if the fungi are connected to several tree species, then I would also not see why they should, in addition, use such a strategy. But I have to admit, it's speculation.
It says, is there any scientific way of testing whether the plant or the fungus has the dominant control of any specific symbiosis? Or is this an unanswerable question?
I don't think it's unanswerable, because if we understand better the molecular mechanisms behind this exchange, then we can probably produce transgenic plants in which we switch off, for example, certain transporters or certain pathways, and then see if we can modify, then we can see who has the control. I think that in the future it will be possible, I believe. Maybe it would even be possible now if somebody has long-term funding and works with Polle, because I think in Polle we could learn a lot about this. It's transformable. It forms mycorrhiza, not only ectomycorrhiza, also avuscular mycorrhiza. I think it can be tackled.
How much do you think that publications like Peter Wohlleben and Susan Simmonard are a danger to science?
I would not consider this as a danger to science. I would consider it rather a danger to society, to be honest. Because what I think is the big problem, if you have a book which mixes, let's say, bits of scientific knowledge with what I would consider fiction, and then use your popularity as an expert to counsel stakeholders. I think there's nothing against this counseling of stakeholders or decision makers, as long as, let's say, we have a balanced composition of such panels. But how should, let's say, for example, politicians know? How should the public know? Because, I mean, if you talk to people, let's say in TV and so on, of course they go and invite people who have this popularity. And therefore, I think it is really very important that we make ourselves heard and that we discuss this. I'm not saying that always, let's say, perhaps for the society, let's say, what we as scientists think is not always the best way for society. I cannot, I mean, this is not what I want to put forward, but I think it is very important that we make our point heard and discussed and valued. As long as we have only, let's say, one view on the world, which is promoted by popular books, this, I think, is dangerous.
Don't you think this is also a thing of where science and society interact, or popularism interacts, and there's two completely different worlds? I think I've never heard of this. Wohlleben rejected a scientific publication as a personal opinion. I didn't know that one. But don't you think this is the real danger, that the public, general public, does not know the difference between what is really somebody's opinion in a book and what a scientific publication is, which is rarely someone's personal opinion, unless it's an opinion paper. It's an opinion at least of several people, normally the reviewers as well.
Yeah, but therefore I said that I think it's not, let's say, in the first place, I would say it's not a danger for the science. I think it's a danger, let's say, for maybe misleading opinions in the public. And of course, then the consequence of this is exactly what you are saying, that for example, when decisions are taken about, let's say, funding or about forest management strategies and so on, they may go into, let's say, wrong direction, in my opinion, or at least could bring us into a very difficult situation. Let's, for example, take this example of Douglas fir or of fir and beech. If we have, let's say, less connected networks, this has some consequences, I think, for carbon storage, carbon sequestration in soil. I don't see it so much as a question of, let's say, trading carbon between trees. I'm very critical about that concept, as you may have realized here. But this for example, in Germany, it is highly, let's say, discussed to introduce Douglas fir as a replacement for spruce. Douglas fir is a non-native species in Germany. What does this mean, for example, for below-ground carbon distribution in soil, for below-ground carbon storage in soil? And so I think this may have really important consequences, and we have to think about this, and we have to investigate it. Because if we would just say it's a non-native species, and therefore we do not want to introduce it here, I think that is a very short argument. But if we have evidence that this disturbs, for example, ecological processes, it would be, in my opinion, a very valid argument. Currently, we do not have such evidence for this, but I think it is something we have to take seriously, and we should investigate it.
This is a challenge for scientists as well, because the popularity of the World Wide Web and the mother tree makes it difficult to avoid just being inflammatory. Do you have thoughts or advice about how to bring up this discussion in non-scientific forums in a way that will engage people in a constructive way?
My practical experience with this is that it's really very difficult. So after our papers appeared, we have been approached by journalists, and we gave interviews. Christian Ammer was particularly engaged in this. And I mean, in the end, the problem is really you cannot convince anybody. So, I mean, we have our opinion, the public has their opinion, and the discussion is often not very constructive. Nevertheless, I believe, regardless of whether the discussion is constructive or not, we have to engage in it, and we cannot, let's say, sit in what we call ivory tower or something. Mm. We really have to take the challenge. I have to say, so after some time, we were really tired of these discussions, because in a way it didn't lead anywhere. So we just had kinds of blocks. Yeah, so one opinion here, the other opinion there. It is really difficult.
Which soil properties are the most important environmental variables for affecting mycorrhizal interactions?
What we found is that let's say the kind of mineral soil plays an important role here, more than the organic layer. Of course, the pH value, because the pH value also, of course, the supply with nitrogen together, they have a very strong influence on what kind of mycorrhizal fungi you will have in the soil, and that, of course, then will have an influence on the amount of carbon and also the processes which you can observe.
What recommendations would you offer to foresters regarding future forest management practices? Should we prioritize planting ectomycorrhizal trees, arbuscular mycorrhizal trees as monocultures, or mixtures of arbuscular mycorrhizal and ectomycorrhizal?
Thank you for this question, because I think this goes also, or takes up, let's say, some other publications which appeared recently, showing that especially the arbuscular mycorrhizal fungi were very important to promote, for example, or to strengthen tree health under high temperature and drought. And if we take this seriously, we have no experience, at least I don't know any publications which did similar experiments here in Europe. So this was done, I think it was done in Canada or the US. But if we take this seriously, I would say it would be a very good strategy to mix arbuscular and ectomycorrhizal fungal tree species and to work with mixtures for several reasons. First, because if you have different tree species, the competition is usually less strong than if you have monocultures. At least for beech, it is known that beech is the strongest competitor for itself, so to say. And so there is a chance for facilitation, and also there is a chance for, let's say, widespread diversity of mycorrhizal fungi, which could prepare forests for different situations regarding, for example, drought, high temperatures, but also perhaps strong variation in temperature between very high and very low temperatures.
I thank you very much indeed for your talk.